Hello, everyone. Five seconds. 10 seconds. That's 20 seconds I can't get back. That's time I could have spent telling you about how 20% of today's fish populations are either extinct or at risk. That's time I could have spent telling you about how cryopreservation may be able to help. Well, let's see if I can make the most of the time I do have left. Cryopreservation sounds daunting, but I promise it's something we're all actually quite familiar with. The basic principle is that at low temperatures, molecular and biological activity either slows down or comes to a complete stop. It's why putting food in a freezer works. Imagine then if we could use a similar technology to preserve endangered fish embryos. We'd be able to build a genetic library and use that information for future research, aquaculture, or even to rebuild entire ecosystems. It is difficult, though, to get the biology of fish embryos to agree with the chemistry of cryopreservation. Cryopreservation requires antifreeze chemicals, and unfortunately, these chemicals are simply too toxic and have a difficult time getting inside the embryo. However, there's a novel class of protective agents called ice recrystallization inhibitors. These novel products work by minimizing how big an ice crystal is able to grow. We hypothesized that these small molecules could be safe for embryos and create a breakthrough in their cryopreservation. Our lab works with zebrafish, and so far we found 3 compelling results. Firstly, we find that getting the inhibitor inside the embryo is as simple as mixing it into the same water. Secondly, we find that exposure to the inhibitor has only minor consequences on the embryo's ability to develop and hatch. Lastly, to determine the cryoprotective potential of the inhibitors, we cooled and warmed the embryos repeatedly to dramatize the size of the crystals. Then, when we looked at the tissue under a microscope, we saw samples treated with the inhibitor did indeed have significantly smaller ice crystals. Folks, today, 20% of fish species are threatened, but in 75 short years, that number is going to be 90%. We have to make the most of the time that we do have left. We can't freeze time, but by supporting this research today, we can freeze the fish. Thank you.